Hi, I'm Andrew, and I'm going to be talking about DNA methylation in under 3 minutes. First of all, many people know that direct changes to DNA, such as mutations, can cause diseases such as cancer. But did you know that changes on top of the DNA can also affect disease like cancer? One example of this is DNA methylation, which might sound a bit complicated because of the name. However, it's not that hard to think of. DNA methylation is when a methyl group is added by methyltransferase, an enzyme, on top of a cytosine base pair, which is the C base pair you learn about in ATCG. So, what does DNA methylation do? Great question. Methylation can silence the gene that it is on top of, which stops the gene from being expressed. You can think of this like in terms of a school librarian. If students are being loud and making lots of noise, the librarian can say shh and silence the students, which stops them from making the noise. So, why does this matter and why should we care? While methylation is normal, cancer can disrupt methylation and silence important genes. For instance, cancer has been shown to methylate and silence important tumor suppressor genes like MLH1 and CDKN2A. When MLH1 is methylated, the cell is unable to produce an important protein, which results in an increased mutations rate that could be up to 100 times greater than normal. Additionally, CDKN2A is extremely important in regulating the cell cycle, as it is a tumor suppressor gene. So when it is disrupted by methylation, cancer can grow unchecked. Now, what are the applications of knowledge of DNA methylation? Well, there's a lot. Some of the biggest ones are for early cancer diagnostics tests. When you can compare data of methylation from cancer and control patients, you can often find many patterns that are specific to cancer or non-cancer people. This could be used to diagnose people with cancer accurately and earlier using patterns in their DNA methylation. Additionally, DNA methylation could be used to identify how people will react to certain medicines, and this way you can create custom medicines for them that will best treat them for their this disease without causing much harm. Finally, DNA methylation could be used in forensics because similar to how people have fingerprints that can be used to identify them, we could potentially look at DNA methylation of bodily fluids or bodily artifacts left at crime scenes and use it to identify someone based on their DNA methylation profile. Thank you so much for watching, and I hope this was interesting. If you'd like to learn more about DNA methylation, you can make a quick Google search or look at my sources, which I've listed below.